Hello everyone, and welcome to our online lecture series for Bio 2. Today we continue on with our investigation of things related to meiosis, particularly focusing on the process of recombination today. Because thus far we've talked a lot about different types of Punnett squares. Hopefully you are familiar with our discussions from last time about topics like epistasis in Punnett squares, where you have a gene that controls another, with one determining coat color and the other determining the ability to make coat color in the first place, with the recessive broken version of coat color turning animals white, whereas the one working copy is sufficient to allow the animal to make coat color in the first place, therefore allowing the other gene locus, the agouti gene in this case for the mouse, to determine whether the mouse is its cute little brown or a black mouse. We've also discussed the ability of these Punnett squares to be carried over through time in a pedigree, looking at how different traits are inherited through generations, or the manifestation of the likelihoods from those Punnett squares, because we do get a ratio, but it's not like that ratio always turns out exactly the way you expect it. We also talked about genes that are inherited on the X chromosome, or other gene interactions like codominance and incomplete dominance, which kind of are the same thing, slightly different in the way that they manifest on the cellular level. And then we talked about the most common type of inheritance, the quantitative, the additive gene, or the additive trait, like height, where we end up getting some sort of normal distribution where we could plot it out as a histogram. So that kind of ties a nice, neat bow around what you've discussed thus far in terms of the lecture and the lab. And today, we weave in the idea of recombination into Punnett squares, ultimately by actually not using Punnett squares anymore. So, one glaring omission from everything that we've talked about thus far is the fact that these genes, these loci that we discuss, whether they be A and C, or genes related to height, or genes related to hemophilia, well, they get inherited on chromosomes. And for example, the human genome has only 23 chromosomes. But we've got something on the order of 30,000 genes. And just so we keep track of this, those 23 chromosomes and 30,000 genes, or rather, those 23 chromosomes are roughly made up of 3 billion bases. So that's our overall genome size. Now, splitting across 30,000 genes into 23 chromosomes, well, you're not expecting there to be only one gene per chromosome, right? In fact, you're expecting there to be something like 1,300 genes per chromosome. And we also actually know that there's some fairly large size variation across our chromosomes. So 1,300 genes per on average, but this here is a human karyotype, where we're able to, using microscopy and some fluorescent probes, identify all of the different chromosomes in a human genome. So karyotype is us laying out all the chromosomes and seeing how similar they are. Well, this particular karyotype has fluorescent labeling, or glow-in-the-dark labeling, on a very particular chromosomal region called an ALU element. This is a transposable element, or a very common genomic repeat. particularly in us humans. And what you notice is that, at the very least, tying this back to our homologous chromosomes discussion, is that you have homologous regions in the pairs of chromosomes that have very similar alu composition, right? You see that for the most part they match. Occasionally, sometimes you get some fairly large structural differences, but that's for another day. But another little side tangent there, sorry about that. But we know that we have these genes distributed across our 23 chromosomes. And because we have way more genes than we have chromosomes, we know that multiple genes have to exist on a single chromosome. Meaning that there are thousands and thousands and thousands of genes on some of our bigger chromosomes, like chromosome 1 here. So how does that work in a Punnett square? Well, we've talked about the idea of independent assortment. Where chromosomes are able to line up independently of one another. Does not matter whether it was mom's on left, dad's on right, or a mix of mom on left, dad on right, mom on right. And in the way of independent assortment, you're able to mix and match. Any gene found on chromosome 1, like let's say the A1 gene versus the A0 allele here on dad. Well, if dads are also B0 and moms are B1, you know that A1 can match with either B0 or with B1, right? Independent assortment says that you are going to create all 4 possible gamete pairs. And there you have it. From this cross here, from this little Punnett square situation, we know that those are the 4 different sets of gametes that you can make. But now what if it looked slightly different? What if instead of lining them up in such a way that you had your A on chromosome 1 and B on chromosome 2, what if A was on chromosome 1 and B was on chromosome 1? Oops. What would that do to your ability to form different gametes? Well, let's draw this out and expand this a little bit bigger. Under these conditions, we would consider haplotypes. And a haplotype is the series of alleles that are connected to one another. So we are always looking at the A and the B gene, and we'll indicate them with a line underneath showing that these are part of a haplotype. And because we see A1 associated with B1, thanks to being mom, we know that the haplotype is A1B1. Our other haplotype is the association between A0 and B0. And in the absence of recombination, these 2 are your only possible gametes that you can make. There is no mixing of A1 with B0 without recombination. Therefore, these 2 types, these 2 different haplotypes are considered our parental type. They are made with no recombination. Now, however, you do know that recombination can occur, and this is the crux of our entire lecture today. Recombination can occur between these genes that are found on the same chromosome, between these genes that are in linkage. They're linked together because they're more likely to occur in a haplotype together. They're more likely to occur in a gamete together. In order for us to break up A1 and B1, we have to have our recombinants, or recombination has to occur. And therefore, these are our recombinant haplotypes. A1 paired with B0, and A0 paired with B1. So in black, we have our parental types, and in orange, we have our recombinant types. Okay, so the theory is there. This is looking at things as a two-point or two-locus recombination problem. And that's what we'll stick to in this class. We won't go further than two, so don't worry about it. Now, why and when do these recombinant and parental haplotypes form? When would you expect to see more parental? When would you expect to see more recombinant? Well, this is where we incorporate math into this problem. So with our recombinant types, we need a recombination event to occur between the region of A1 and B1. So let's go ahead and grab this and blow it up a little bit. We need some sort of recombination to have occurred somewhere in between the space between A1 and B1. Now, I don't know how likely that is, but I can look at progeny ratios and find out. So ultimately, we're going to be looking at the number of children generated with either parental or recombinant types to determine our recombination distance or recombination frequency, meaning how often do you get some sort of recombination event that switches these two genes around. Now, I do want to make one simplification to our drawing here to keep things a little bit more consistent. Instead of going across the centromere, because it will bother me, we'll assume that these two genes are now actually on the same side, the same chromosome arm. So just one aesthetic change, not really anything that you need to worry about till way, way later in your biology education, but I don't want to be drawing things kind of crazy. Same chromosome arm. And that has to do with the fact that we expect one recombination event per chromosome arm. Makes it a little bit more realistic to be calculating recombination frequency between genes whose loci are found at least on the same chromosome arm. So, what's the likelihood of a recombinant occurring between these two locations? Well, we'll report that recombination frequency as a genetic distance. And genetic distance is measured in the unit centimorgans. And yes, to differentiate this from centimeters, it is abbreviated as lowercase c, capital M, centimorgans. And centimorgans are essentially a percent recombining. So, in a hypothetical situation, if I have 100 offspring, and I notice that 80 of them are the parental types, meaning in our example here, either the A1, B1, or the A0, B0. Those are our two parental types. And that would leave 20 as our recombinant types, meaning that they are either A1, B0 or A0, B1. If we see that there are 20 recombinants to every 80 parental in 100 offspring, we can calculate centimorgans as the number of recombinants divided by our total offspring. Well, in this case, we had 20 recombinants over a total of 100, which gives us a recombination distance of 0.2, which gives us a recombination frequency of 0.2. Turning that into a percentage, we have 20% recombination frequency, therefore we have 20 centimorgans. So I should rather say this is times 100 in order to convert to centimorgans, because you want to convert this ultimately to a percentage. So we can report the genetic distance between the loci A and B as 20 centimorgans, because in 100 offspring, we see 20 of them have experienced recombination, or a break that mixes and matches A1 with B0 and A0 with B1. Now, recombination is only detected here in this cross where we have the 4 different alleles. So you're oftentimes going to be using something like a dihybrid cross, or a lot of hybrids in determining genetic distance. But it does not matter whether the A1s and B1s are on the same connected chromosome. The reason why I'm pointing this out, even though that feels intuitive, is as follows. One of the most common mistakes is making that assumption about connectedness based on capital letters or numbers in terms of alleles. So let's go ahead and draw some chromosomes out. These are going to be a little bit cartoonish in the sense that it's really long arms with really short arms on the bottom. And we can see that we have the loci A and B on each of these 2 homologous chromosomes. Now we painted them as pink and blue to indicate parental origin, or that this was derived from the original mother and this was derived from the original father. But what you really care about is which 2 alleles are connected on haplotypes. Because I could have, for example, A1 and B0 and A0 and B1 connected on haplotypes. This would have me draw my parentals like this, P for parental, with that underlining indicating haplotype, and our recombinants here as A1, B1 because if I want to make one type out of this, oops, too big. If I want to make one haplotype out of this, I'll start reading from the top, have the A1, then I will break off somewhere in the middle and read off to the B1. And then the remainder of the chromosome is actually derived from the B1 side. So drawing that in terms of our colors, it would go something like mom, break to dad's, chromosome arm, and down. And this would be our recombinant type that matches from our two different parents. Even though now the A1s and the B1s and the A0s and the B0s are on the same haplotype. So a little bit counterintuitive there. Now why, when I did the 1 for A1, B1, did I also include A0, B0? Well, going from the other side, if you had a recombination event that occurred here, there would have been one gamete made starting pink, ending blue, and there would have been another gamete made that starts blue, converts over to pink, and continues on as pink. So, because we assume that you make the one, we also assume that the other is produced, which it has to be in order to make the one in the first place. So, that digression should convince you that parental and recombinant do not have anything to do with the actual alleles present, whether they be capitals and lower cases or numbers, but instead they have everything to do with our parent of origin. And so in many of these problems, when I'm discussing recombination frequency, you're going to actually start out with the parental generation. Hence why we call them the parental types. And in that parental generation, you will often encounter true breeding parents. Meaning that mom may be A1, A1, B1, B1. When we go ahead and draw out her chromosome for this, or her two possible chromosomes, we know that in every single case, because the A has the same allele and the B has the same allele, that A1 will always be connected to a B1, and that A1 will always be connected to a B1. So who cares what chromosome it derives from? Mom is always going to be donating these two alleles, meaning that the parental generation is oftentimes homozygous. Now you are frequently going to be taking this mom and crossing her with a homozygous of the other allele dad. Because in this case, where you have all homozygous at all four of these on dad, you are also going to get obligatorily A0 linked to B0 and A0 linked to B0. Now, when these cross, you know that A1 will be linked to B1 and A0 will be linked to B0. In the filial 1 generation, we now have a linked hybrid. And the linkage is determined by the types in the parents. I could have just as easily gone back and said that A1 was linked to B0 and A0 was linked to B1. This would have changed what was present down here by connecting A1 with B0 in the parental and A0 with B1 in the other parental. So you need to look and see what alleles are linked in the parents before you look at how they manifest in the hybrid. And now, within the hybrid, you are able to measure recombination. Because you have different alleles, you have something called a genetic marker. We'll talk more about genetic markers later, but genetic markers are a way for us to distinguish maternal versus paternal strands. So whether they derive from mom or dad, or who they derived from, or the fact that they are different in the first place. These markers are fundamentally something about the DNA sequence. So once we have those markers, we then are going to self or cross this individual. And there are two different ways to look at this. You can perform something called a test cross. A test cross is just a way for us to determine exactly which gametes came from our hybrid. So therefore, a test cross is crossing your hybrid with a homozygous individual. Meaning that they would be, let's say, A0, A0, B0, B0. And then you go ahead and look at their progeny. And you're going to be able to create only a couple of different progeny here. In the case of the test crosser, the homozygous individual, they'll only donate an A0 and a B0 allele to the child. From your hybrid, they will either add an A1 B0 as a parental, an A0 B1 as a parental, or a recombinant type that is A1 B1 and a recombinant type that is A0 B0. So then you can mix and match these. If I am pairing my test cross with this parental, I am going to pair my test cross again with this parental. I am again going to be pairing my test cross with this recombinant and my test cross with this recombinant. These are your 4 possible progeny made from this test cross. Now, you could additionally, and probably should, recognize that these should be written A1, A0, B0, B0, A0, A0, B1, B0, A1, A0, B1, B0, and A0, A0, B0, B0. What you will notice here is that your parental types are the ones that actually have a mismatch in the alleles, because the original parental types also had a mismatch in the numbered alleles here. So do not think that because my recombinants or I seem to have more matching with A1s and B1s and A0s and B0s, or all B0s here, do not think that those are your parental types. These two at the bottom are your recombinant types because they reflect the inheritance from the parents. So hopefully this has made sense to you as I've explained it to you a couple different ways about tracking recombinants through the parentage. So in doing this test cross, where we are crossing a hybrid against a homozygous individual, we then can look at the frequency of progeny made. So let's go ahead and say that I had 100 of these, 100 of these, 50 of these, and 50 of these. So, if I want to calculate my recombination distance, what I'm going to have to do is put my number of recombinants over the total. So, what is the total? Well, 100 plus 100 plus 50 plus 50 is 300 individuals. What is the total number of recombinants that I have? 50 plus 50 is 100, meaning that we have a recombination distance of 0.33%. Converting that into centimorgans means it's 33.33 continuing centimorgans. And so that is the way to calculate a recombination distance using progeny and a test cross. You identify which progeny were made from our parental types in our hybrid and which progeny were made from our recombinant types in our hybrid. Alternatively, rather than performing a test cross, you could also genotype the gametes. Meaning that you're going to your hybrid individual and sequencing their sperm or eggs. And most of the time, because of abundance numbers, you're looking at sequencing the sperm. You will therefore only get haploids, but you can still see whether the counts match the parental types. So you'll be able to go in and count how many sperm have A1B0, and if we again have that number at 100, how many sperm have A0B1? Go ahead again and put that number at 100 for the simplicity of math here. And then you're going to look at your recombinant types, A1, B1, and A0, B0. These would again be 50 and 50, keeping consistent with our previous example of the test cross. You again have your number recombinant over your total. The total is again 300. The number recombinant is 100, giving you the exact same recombination distance. So what can I do to make this more complicated when you're solving things? Well, I won't be writing these in the parental recombinant order, right? No purpose in doing that. I want you to be able to track from parentage what the different recombinant totals are. I also will be changing up the numbers. They won't always add up nice and neatly, and I will be changing the recombination frequencies. So let's take a look at an example problem where it's not done in a way that's particularly neat for your understanding, but instead something that we have to pull apart. So, new space, example problem time. So we're going to revisit our epistatic control gene here. Not that it really matters that the gene's epistatically controlled, but it allows us to talk about an example that we've had before and also switch our nomenclature. So with our epistatic gene, you note that there are two loci. one that determines the color, and there are two alleles for it, either agouti or black. This is our tan color, or the agouti color, and this is the black allele. And then there is the control. We're going to label it our epistatic controller. And the epistatic controller is either on or it is off. The two different alleles here. And if you happen to be CA, CA, EO, EO, you make pigmentation, and the pigmentation that you make is our agouti tan. So just a reminder of the functionality here. So that gives you a setup for what the actual genes that we're looking at here are. But let's go ahead and look at this in terms of recombination distance. Because in this situation, we know that the epistatic controller and the color loci are actually linked. And so we're going to determine the genetic distance between these two loci by performing a couple crosses. So we're going to start out with an individual that is white, but we know that their color loci are also homozygous. It's homozygous at the epistatic controller, therefore they're white. But also that at their color loci, we know that they would make the black pigmentation if the epistatic controller was on. So we have one parent that is CA, CA, EF, EF. And we need to cross them against something that is agouti and homozygous, and the epistatic controller is on. Now, this means that the two connected haplotypes, as we draw them out here, are CA alongside EF and CA alongside EF. And that the two haplotypes here are capital CA, sorry about that, don't make those mistakes, our capital CA, EO, connected to our second CA, EO. Now crossing these two, the only possible combinations that we can make are of a CA with an EF and a CA with an EO. This is the F1 hybrid. In our F1 hybrid, we're then going to go ahead and allow meiosis to occur. And after meiosis, we're going to genotype the sperm. And see how many of our parental type CAEO are made, how many of our parental type CAEF are made, and then calculate or count how many of our two different recombinant types there are. So what are our two different recombinant types? Well, C capital A with an EF and C lowercase a with an EO. And so from these different types, cut off the top of that E, we can now report our sperm counts. In these sperm, we see that there were 104 sperm counted as CAEO, 93 sperm reported as CAEF, 21 sperm reported as CAEF, and 25 reported as CAEO. EO. Our total for this is 104 plus 93 plus 21 plus 25, or 243 total sperm. In order to get our recombination distance, we have to take our total, 243, and use that number to divide our number of recombinants. Our number of recombinants are 21 plus 25, or 46. So 46 divided by 243, well, make sure I enter that in the calculator right. We get a recombination distance of 0.189. We know that we can convert that into a centimorgan by moving the decimal place over 2 points or multiplying by 100, same thing. So we get 18.9 centimorgans, genetic distance between our color gene and our epistatic controller of that color gene. So hopefully this makes some intuitive sense for you in order to be able to calculate these recombination distances for two different loci on the same chromosome. When we have recombination distances for multiple different genes on the same chromosome, because remember, there are several hundred, even several thousand genes per chromosome, we can take all of that information to create something called a genetic map. And a genetic map is just all centimorgan distances between genes on the chromosome. And you'll notice that maybe if you're looking at a chromosome here, that there may be one gene here, one gene here, one gene here, one gene here, and that this distance is maybe 18.9 centimorgans, this distance is 25 centimorgans, this distance is 5.2 centimorgans, and that'll get you thinking. Oh, actually we'll add one more here to the one that's really far down. 33 centimorgans. What is the actual distance? If these genes are A, B, C, D, and E, what is the centimorgan distance between A and E? Well, you could add up 18.9 and 25 and 5.2 and 33 and get a centimorgan distance, and that's how you report it on your genetic map. But my question for you is, what is the true recombination distance between A and E here? Or, what is our maximum recombination frequency? What is the upper limit on centimorgans? And this is real, meaning in terms of in practice, not in terms of what we can calculate, because you can calculate and report a centimorgan distance higher than this upper limit. But in all reality, the likelihood that a gene is quote-unquote linked to another gene, or the likelihood that they occur on the same quote-unquote haplotype, even though they're not on the same piece of chromosome, is 50%. Because you have a 50% chance of lining up even if you are on opposite chromosomes or on other chromosomes. So because there is this 50% recombination limit in terms of, or 50% linkage limit in terms of likelihood of A1 being paired with B0 or likelihood of A1 being paired with B1, while they're on separate chromosomes, the true recombination distance that we'll be looking for, the upper limit, is 50 centimorgans. So you'll never get a value between two loci that is higher than 50 centimorgans. Because even if they're on separate chromosomes, you'd still get that value of 50 centimorgans in the recombination distance. This means that if you are calculating recombinants that end up giving you more than 50 centimorgans, you've identified the wrong recombinant haplotypes. I'll say that again. If your identification of parental and recombinant haplotypes gives you a recombination distance greater than 50 centimorgans between two loci, you have misidentified the recombinant types. You will always have fewer than 50, or always have fewer or equal to numbers, with a strong bias for fewer, recombinants than parentals. Because remember, parentals are made without recombination having occurred. Recombinants are only made when recombination occurs between that spot. But recombination can occur in many different locations on that chromosome. So most of the time, you should be looking at the lower of the two in order to identify your recombinant types, and the higher of the two in order to determine your parental types. So I will be, I'll take a second here to give you guys a couple of other problems, so that way you can take a moment here and practice calculating recombination distance between these different loci. So all three of these are going to be following a similar pattern where we have the A and the C control and the color and the epistatic controller loci. And in these different cases, we'll have different connections between your haplotypes. So, in the first, your parent 1's genotype is C capital A, EO, EO, and your parent 2's type is C lowercase a, EF, EF, just as we had observed. Now, you are getting in the sperm counts CA, EO as 64. CA, EF as 30. C lowercase a, EF as 61, and C lowercase a, EO as 33. Whoops, have to be able to read my terrible handwriting. So that is one example problem. I'll put it in a blue box here. Can't even draw the box. There we go. For a second problem, you now have the different parents in that it's CA, CA, EF, EF for parent 1, and parent 2 is going to be C lowercase a, EO, EO. Here, you do the sperm counts for your 4 different sperms. Let's write them out. CA, EO, CA, EF, CA, EO, CA, EF. And the numbers for this group are 6 of these, 144 of these, 150 of these, and 4 of these. Problem 2, outlined in the green box. And for our last problem, we'll have the exact same parentage as up here, with the numbers being slightly different. So we'll copy this set of numbers, or that set of haplotypes in sperm, and instead report our numbers as 226 of these, 227 of these, 64 of these, and 66 of these. So same parentage, so I'm going to put it in the same box. But please go ahead and calculate the recombination distance for these problems. Additionally, you can go ahead and make up some of your own and exchange them with your friends. Once you understand how to do them, I strongly encourage you to start making up your own. That shows true understanding of this process. And our next lecture is actually going to build on this observation by using some statistics. So it is absolutely imperative to move forward that you understand how to do these 2-point test crosses to determine recombination distance between two loci. But that's enough with the math today. We'll end the lecture with a brief discussion of some biology and chromosome structure by discussing just a few more terms. So the first term that I want to talk about is synteny. Synteny is our way of looking at preserved gene order in distantly related species. And it's our way of looking at how the genome has restructured itself through time. For example here, we have two different organisms, a mouse and a human. Mice have 19 autosomes, humans have 22 autosomes, so going up to 19, up to 22. And then we have our sex chromosomes of the X and Y. Mice also have the X and Y. And what you'll see here is chromosome painting, where we've taken chromosome 1 in mouse, painted it red, and then in the human genome, painted all of the syntenic regions in red, meaning anywhere that you find blocks of genes that are identical in human chromosomes, you will paint them red. So you see that there's some chromosome 1 here, there's some chromosome 1 there, chromosome 1 seems to be dispersed here, chromosome 1 seems to be dispersed there, and even there's a little bit of chromosome 1 over here on chromosome 18. And in this way, you can see that chromosome 1 of humans and chromosome 1 of mice are not sharing complete identical history, but rather there's been massive chromosomal relocations. We see those chromosomal translocations, inversions, deletions, and duplications genome-wide. And you see that this is true for all of the mice chromosomes 1 through 19. We again see a large block of chromosome 2 in orange, and chromosome 2 in humans does have some orange, but the rest of the orange is dispersed across other chromosomes. In fact, chromosome 20 seems to be a whole piece of chromosome 2 that's just maintained on its own. So in this way, we can piece together the history of chromosomes by looking at gene order across distantly related individuals, or distantly related species. Now you will note which chromosome seems to have undergone the absolute least rearrangement. We'll look at that X chromosome. look at how similar. There's no insertions from other genes, from other chromosomes rather. The X chromosome in humans is just large blocks of shared synteny with the X chromosome in mice. So our sex chromosomes don't seem to rearrange much, but our autosomes rearrange a whole lot. So this is why we make recombination maps for different organisms. Now, this history of genome restructuring can be analyzed by synteny, but also by looking at the process of fractionation. And fractionation is gene loss after some sort of whole genome duplication event. Meaning that at some point in the meiotic history of a species, it retained a lot more of its genome, or a local chromosomal duplication. After fractionation, you usually get diploidization, meaning that you start losing some of that double genome piece by piece. So let's take a look at this really cool diagrams from the corn world from one of my former collaborators, Jim Schnabel. So this illustration shows that the ancestor had all of these genes. It underwent a genome duplication, so it doubled its entire genome, as plants tend to do. But then over time, you didn't need 2 backup copies of everything. You might have accidentally deleted the teal gene in sequence 1 or chromosome 1, but retained teal in the copied chromosome 1. This will repeat for many different regions. You can study those that are retained in both, or you can study those that are lost. Now, the last bit of vocabulary that we want to lay on our study of syntalogs here, because these are syntenic orthologs, so genes that have synteny with each other, plus shared sequence or shared ancestry, are syntalogs. Genes that are copies of one another but don't have synteny, so without the syntenic analysis, you can see 2 copies of the blue gene in both in this single individual. So 2 copies of same gene in one species. Sorry about that. Hate clicking that button. In one species, because they are evolving in parallel to one another in the same species, are called paralogs. When I have 2 copies of the same gene in different species, like looking at the gene here in mice and say that this region happens to be syntenic with this spot right here in humans, these 2 that share history are called orthologs. So, if you are evolving in parallel because you're in the same genome from a genome duplication event, you are paralogs. If you are evolving in different species from a shared history, you are orthologs. And if you are a type of paralog that is evolving in the same syntenic block, so a whole genome duplication, Jim Schnabel has deemed you a syntalog. And we can see that this synteny map, and this is kind of a complicated image, but just notice the connecting lines. In the middle, you have the grass sorghum bicolor, so sorghum. And on the top you have one region of corn, and on the bottom you have a region of corn. And you can see the duplicated history and the subsequent fractionation that has occurred. Now, in the top region of the maize genome, you see that it's retained this first block of very important gene up front. And it's also retained some of this stuff in the middle. And it's also retained this gene here. But it seems to have lost this component. That doesn't exist there anymore. It does have that endpoint. So we see that this is an area of loss, and we also see that this back end block that should be there is also a loss. Now looking at the region below, you see that the second region of corn has been lost here. Does not have that chunk of a gene. You notice that it has retained these pieces, whereas this piece is actually a loss up here. You do notice also that the back end is shared quite completely. But that back gene block is not. And so you can, as a biologist, start studying this history to see which genes are preferentially retained and which genes are not. Which genes do you need 2 copies of, and which genes can you get by with only 1? And how does evolution shape those different genes as to what they do? So these will be some vocabulary questions on your quiz for this week. But please do expect that you will be getting some recombination questions, simple recombinations, on the quiz this week. And nail down this process before we get into the statistics for next week. We will be wrapping up our discussion of Punnett squares, inheritance, chromosome linkage, with a discussion of whether I am telling you the truth or not about genetic distance using the chi-squared test. See y'all next time.